This is the fourth lecture on early season apple diseases, and we'll focus on powdery mildew. The learning outcomes for this lecture are: 1, describe the signs and symptoms of powdery mildew on fruit and foliage. 2, describe the life cycle of powdery mildew and the conditions for disease development. And the final outcome is to be able to identify management strategies that are used to control powdery mildew on apple. Powdery mildew is caused by the ascomycete fungus Podospharia lusotrica. The fungus overwinters as mycelium inside buds that were infected in the previous season. The fungus can infect all types of tissue, including buds, flowers, leaves, shoots, and fruit. On all of the tissue types except mature fruit, the fungus appears as white tufts that can coalesce and cover the entire surface of the tissue. So on panel B, you can see a single lesion. In panel C, you can see that the lesions are coalescing to cover the leaf. And in panel D, you can actually see some of the leaves are completely covered with powdery mildew. On some varieties of fruit, symptoms appear as netting, russeting, or ghost spots, as you can see in E and F. So E, this is showing a symptom called ghost spots, and F, this is russeting, or sometimes also called netting. Powdery mildew interferes with the proper functioning of leaves. It can reduce shoot growth and also reduce fruit set. Infections can also cause deformed shoots, flowers, and fruit. Like apple scab, powdery mildew is a polycyclic disease. Cnidia produced in the spring from overwintering mycelium serves as primary inoculum and initiate primary lesions. These lesions then produce more cnidia, which serve as the secondary inoculum. Infections occur at temperatures between 65 to 80 degrees Fahrenheit when relative humidity is high. However, free moisture is not needed for spore germination to occur. It is not surprising that the temperatures that favor initial infections are similar to the temperatures that favor bud break. Buds can become infected as they begin to form until they are matured for overwintering. So this would be at the overwintering stage. This has a chronic effect on the tree, reducing tree vigor and fruit yield over several years. Ascospores do not play a significant role in the infection cycle. This is because the Chasmothecia, which is shown, there's one shown here, which is a Chasmothecium, previously called Cleistothecium, do not split open easily, and when they do break open, the majority of the ascospores are immature and don't germinate. Cnidia form in chains on the lesions, as you can see in the image on the left, and these spores are easily dispersed by the wind. If Chasmothecia form, they form within the lesion, and they usually do not produce mature ascospores. So in these images, you can see the Chasmothecium, and you can see the asci, which have very few, if any, in this image, mature spores. So buds infected with the powdery mildew fungus are less winter hardy. They break later than healthy buds and are often malformed, as shown with the bud on the right in this image. So this healthy bud has already emerged, and this infected bud emerged later or broke later, and you can see that the tissue is malformed. By breaking later, the fungus will have healthy green tissue available for new infections. In warm production areas that don't have cold winters, powdery mildew is a major disease because the infected buds survive the mild winters. The image on the left shows a close-up of a sporulating bud with chains of conidia. So you can see the chains of conidia here. And this often can be seen under the dissecting microscope. A compound microscope isn't needed. Spores from the sporulating buds are wind dispersed, as I mentioned, and these are the ones that initiate new infections. Flower clusters that are infected, as shown on the right, are smaller and bloom later than healthy flower clusters. It's also not uncommon for a twig to have clusters that are healthy and infected. So on this twig, you can see that there's an infected cluster here on the right, and on the left, there's a healthy cluster. You can also see that the healthy cluster is at the pink tip stage, whereas the infected cluster is still at the tight cluster stage. You can also see in the background here that the twig has powdery mildew on it. So conidia produced from primary infections serve as secondary inoculum and can infect new leaves, shoots, and to a lesser degree fruit. Infected flowers shrivel and die, and as a result, powdery mildew infected trees produce less fruit. Leaf and shoot infections can continue as long as shoot growth continues, which generally corresponds to 1 to 2 weeks after petal drop, depending on the variety and the temperatures. And these images just show different stages that are infected with powdery mildew. So you're at a pink tip with this flower cluster. This flower is shriveling and dying, and this is a flag shoot, and you will continue to get these flag shoots until the shoot growth stops. Integrated management of powdery mildew includes the use of resistant varieties, cultural practices, fungicides, and or biocontrol products. Management targets reducing the primary inoculum in the orchards and the spread of the primary and secondary inocula. Management of powdery mildew begins during the dormant season and continues through harvest, as shown by the gray stars on the disease cycle. So you're going to be initiating management down here at the dormant season, and you're going to be continuing all the way through to harvest. Resistance to powdery mildew is present in several varieties, including some of the more popular varieties such as Gala and Fuji. If susceptible varieties are planted, we recommend that growers interplant different varieties to have a spectrum of resistance within the orchard. Interplanting can help control primary and secondary inoculum levels in the orchard. Growers should also consider the growing conditions in the region that they are producing when selecting a variety. Growers in regions that have mild winters should consider planting resistant varieties, since the pathogen can survive in the buds since they don't have hard freezes in these milder regions. So dormant season pruning to remove as many of the infected buds as possible is critical to reducing inoculum levels in the spring. This is especially important during mild winters. The debris that's pruned off should be removed from the orchard and either burned or put into the trash. Although Cleistothecia aren't normally produced, and you don't often get mature ascospores, we still don't recommend composting of infected tissue. So the application of fungicides begin early in the season and will reduce the amount of primary inoculum that can be dispersed through the orchard. Applications of fungicides occur on a 7 to 10 day schedule, beginning at bud break and continuing to the end of terminal shoot growth, which is around June in central Ohio. Once temperatures get above 85 or 86 degrees Fahrenheit, fungal growth is inhibited and the need for powdery mildew specific fungicides is less important. Contact fungicides such as sulfur are very effective. They also are cost effective in that they cost significantly less than some of the eradicated fungicides. Smothering products such as oils can also be used to prevent the dissemination of Canidia. Smothering products, as well as biocontrol products, should be applied after infections are first observed, as they have no efficacy as protectants. So the next lecture will be the final lecture that covers early season apple diseases, and in that lecture we'll cover juniper rust, focusing on cedar apple rust.